Hi everyone, welcome to this episode of Kuiper Labs. In today's video we're going to be comparing the processes of mitosis and meiosis when thinking about the way that cells behave and also what happens to the chromosomes that exist inside cells during these processes. Okay, so let's do a quick little recap, or perhaps this is the first time you've even heard these words, in which case all the more important that we do a quick overview of them. Okay, so mitosis and meiosis are processes of cell division. Okay, so we're thinking about cell division. Okay, so using these processes where existing cells divide and then multiply, so divide in order to produce more new cells. Okay, so mitosis is the process of basically like cloning. Okay, so making copies, so identical copies of existing cells. Okay, and that we use mitosis when we're thinking about growth and repair. Okay, so building new cells of the same type to make you larger, or to make things bigger and stronger, or repair in order to replace or fix up any damaged tissue. And then meiosis is cell division, but with the specific purpose of forming sex cells, also known as gametes. Okay, that you may remember that term when we've looked at reproduction in the past. So sex cells being sperm and ova, the plural of ovum. So ova being in the ovaries and sperm being in the testes in males. Okay, so that it's specific formation of sex cells. Okay, and so the idea being that that these cells are supposed to be different to existing cells. Okay, so we're going to go through some of the processes involved in each thing, and then we're going to put together a table at the end to kind of compare them. Okay, so here we go. We're starting with our original cell. So what we have here in this existing cell, we've got one pair of chromosomes. Okay, so just to keep it simple. Okay, so it's got a green and a red. So what happens inside this cell is that then each of these existing chromosomes multiply. We say that it replicates itself. Let's see if I can write that here so you can see it. Replication. Okay, so what that means is that inside our cell over here, we've now got two green strands and two red strands. But rather than be kind of like separate like this, what we see is that actually they end up being connected like this into this kind of X shape. Okay, now so what then happens is that then they start to move up into the middle of the cell, which then the main kind of body of the cell, if I clean this up a little bit to make it a bit easier to see, starts to elongate. Okay, so we get this one in the middle here, this one in the middle here. And what we have, if you forgive the extra kind of colours, is that we kind of get these little kind of fibres that attach to the ends here, which kind of join on to these chromosomes and then start to pull them apart. So then we end up, so we've kind of got this sort of shape as the cell has started to pull apart and then kind of split itself into the middle. Okay, and so we end up with a green and a green, a red and a red, still being pulled towards the ends. Okay, and the rest of the cell is dividing, and so we end up kind of splitting into the middle, and then we have a green in each and we have a red in each. Okay, now there are names for each of these processes that you totally don't need to actually be able to identify. The key things that you want to be able to see here is that we've got two identical, what we call daughter cells. So they're the ones that come as a result of the parent cell over here. Okay, then you see that these are identical to each other, and they are identical to the starting cell. So this is why we call it cloning. Sorry, this is cloning in inverted commas. Okay, as far as we've actually made an identical copy, where we had one cell before, we now have two, which is why we think about it in terms of cell growth and repair. Okay, because we can make more cells, or that we can use them to repair or replace damaged cells. Okay, now we're going to have a look at meiosis, which has a lot of similarities. Okay, so here we have the same kind of starting point as we did in mitosis. Okay, so we're taking this time that then we get replication. Okay, so that each of these chromosome strands has doubled itself, and that doubled one is kind of connected there. But now what happens is that now we get something a little bit different. Is that rather than these kind of lining up in the middle, maybe if I draw them this way, you might be able to see it a bit better. So rather than them purely lining up across the middle and then being split in half like we saw just before, what we see is that actually one whole pair goes to one side and the other whole pair goes to the other side. So if I draw it a bit more like this, okay, so we get the green set there and the red set there, and they're being pulled towards the end. And so what we end up with is we end up with two daughter cells in this first stage. This one's got our green pair in it, and this one's got our red pair in it. And then what happens is that then we undergo the same sort of division process again. So a second division process. Okay, so where we've got our red and our green, and then we're being pulled from each end. We divide in the middle again, and so we produce 4 daughter cells. Like this, 2 like this, and 2 like this. Okay, now I'll see if I can bring that down a little bit just to help you to see this a little bit better. So the idea is that we've produced 4 unique daughter cells. Now you see that over here that we had 2 chromosome strands. Each of these only have 1 chromosome strands. That is, each of these daughter cells has a half set of chromosomes. So 23 versus 23 pairs that we had in our original cell. Okay, so there's 2 key differences in that to mitosis, and we'll outline some more differences in a sec. That we've ended up with twice the number of cells at the end, 4 instead of 2, but that the cells that we have only half the set of chromosomes. Now, what the reality is that this makes it actually look a little bit simple. I know scientific ideas don't. You may not be thinking this is very simple, but what the reality is that these 2 things aren't always exactly the same as each other. These 2 things aren't exactly the same as each other. In this process of these kind of things pairing up and being pulled apart and so on, that often there's a chance where genes can cross over from one strand to another. And so we can actually end up with, this might be green with a bit of red here, and then this might be green with a bit of red here, and red with a bit of green here and here, so that we're actually kind of mixing and matching of genes, which is one reason why you have features that are similar to your parents, but you aren't a clone of your same-sex parent. I'm not a clone of my father. My wife isn't a clone of her mother. Okay, that the idea is that you don't end up, it's just a mini kind of clone. Okay, so now we're going to take some time, we'll quickly sketch up a little table of some similarities. Okay, so I want you to sketch up a table like this in your book. There's going to be a list of about 5 or 6 things in each thing, so make sure you leave some space here. Okay, so to start with, with mitosis we end up with 2 daughter cells, whereas in meiosis we end up with 4 daughter cells. Okay, our two daughter cells are genetically identical. Whereas here in meiosis we have genetically unique. So distinct from the parent cell and also distinct from one another. Okay, so genetically identical, genetically unique. We have one stage, and here in meiosis we have two stages of division. Okay, so we saw in mitosis it divides once, in meiosis that it divides twice, two different stages. Okay, so in mitosis we end up with 23 pairs of chromosomes in each daughter cell, whereas here we end up with 23 chromosomes. That is half set. Okay, and then we have a technical name for this. Over here we call the cells that we produce diploid for two, di for two, and here we talk about haploid, which is for half. Okay, two for double and half. And so what we're talking about here, we talked about growth and repair, and here we talked about sex cells, that is gametes. Okay, so you can see that these two processes have a lot of similarities in terms of that we're involving cell division, but they're not the same processes. Okay, that then so that we're producing a different number of daughter cells that have different characteristics in terms of their genes. So what are the chromosomes like? We've got twice as many stages of division in meiosis as we do mitosis. We end up with cells that have the same number of pairs of chromosomes, 23, in mitosis, but only a half set in meiosis. And then accordingly, we use the terms of diploid and haploid to talk about those types of cells. And we also use these processes in different parts of or different aspects of how your body functions. So for growth and repair, and also just kind of normal kind of, and we can also would say asexual reproduction. So that when we're thinking about, say, grass kind of growing or sending off runners, or a tree kind of growing, that's this idea of asexual reproduction, whereas here we're using it in sexual reproduction. Okay, so that there's a different outcome, there's a different use and purpose, so we need different cells to meet that. The reason that we only want a half set of chromosomes in each of our sex cells is that we have to have a sperm and an ovum meet. So we have 23 in one plus 23 in the other leads us to 23 pairs in our new zygote at the other end, which is the whole idea of that. Okay, so in this video we've outlined the stages in mitosis and meiosis, and then we've spent a bit of time comparing the two things. Okay, going through some of their features and ways in which they're similar and different. All right, thanks very much for watching. Bye for now.